Well, it's really wonderful to be here, even though I'm only 45 minutes away. I've never been to this amazing place. So thank you so much for inviting me. And I know I'm going to learn a ton from this meeting. So I'm going to tell you about our work trying to use big data to actually, to be honest, transform healthcare. That's our mission. And I'm going to start with a premise that I think many of you will agree with. Some of you may be offended. But I would argue the healthcare system is, I used to say completely broken, but now I'm toned it down a bit. I say flawed. And that's true in many, many different ways. And this just illustrates some of those. First, we're very focused on treating people when they're ill. We're, as such, it's very reactive. When people are healthy, we measure very few things. And I would argue some of those things aren't terribly useful. And yeah, the frequency when you're healthy is usually very rare. If you're a healthy person, you don't go to the physician very often. But the biggest problem is the one at the bottom. Nearly every decision about your health is made on population-based measurements. I think we would all agree we should really be focused on keeping people healthy. As such, be very proactive. We're certainly capable of measuring many, many more things. And I would argue the frequency should depend on what you're at risk for and what your personal trajectories look like. And then lastly, I think we would all agree it should be individual-based medicine rather than population-based. Most physicians will say we do that already, but I would argue it's not at the level at which we'd like to see it. And so we should really be focused on precision health rather than precision medicine. So to illustrate this point, I like to show this slide here. We've been taught since we were little that your temperature, when you put a thermometer in your mouth, is 98.6. But if you look at the studies out there, actually, first of all, that number is usually not right. The median is actually more like the one here, 97.5. But more importantly, there's a spread. The 25th quartile is 94.6, and the 75th quartile is 99.1. So what that means in today's world, if your baseline is 94.6, and you go to a physician's office and they measure you at 98.6, they'll tell you you're healthy, everything's fine. What are you doing here? But if you're up 4 degrees over your baseline, the odds are you're rather ill. And so that's a theme we hammer many times. It's really important to know your baseline so you can see that change from that. So a number of years, there are many things that affect your health. This just shows some of them. And obviously this meeting covers a lot. Your genome is one part of this, but your activity, lifestyle choices, meaning the food you eat, stress, environmental exposures, these all impact your health. And we're in a world where it's now possible to quantify all these, some well, some not so well. Activity you can quantify decently. Food you can do, but it's pretty clunky. And environmental exposures I'll spend some time on today. But even if you can't quantify these things very well, you can quantify their effect by making deep molecular measurements on people. And so a number of years ago, we set up something we call personal omics profiling on a rather small cohort, 109 people. And we've been running this study for about 8.5 years. I was the first participant, so for me, it's about 11.5 years. Some have been a little bit longer than 8.5, some a little bit shorter, but that's the median. And basically what we've been doing is actually we sequence their genome once, and then we do these deep omics profiles, if you will, while people are healthy. So one aspect is big data. We're actually collecting data mostly off peripheral blood monocyte cells for the epigenome, transcriptome, proteome. Some of it's out of plasma, for example, the proteome, metabolome, lipidome. We measure people's microbiome. So we now have gut, nasal, and recently we've completed the analysis of skin and tongue. That's actually quite interesting. We do deep clinical testing, other kinds of measurements as well, some mental health types of measurements as well, though we haven't analyzed all those data yet. We do some advanced tests like stress echocardiograms and a lot in the area of glucose control, as you'll see in a minute. And about 8 years ago, we added on wearable devices so that we can track people continuously. And I'll talk quite a bit about that. So one aspect is again, deep data collection on people. And the other aspect is the longitudinal profile. We sample people every 3 months while they're healthy. And if an adverse event comes along, like a viral infection, other sorts of things, accidents, what have you, we'll take many more samples, typically 5 to 7 additional samplings, including immunizations. We'll sample through that as well. So again, it's a matter of deep measurements and following people over time. And you might say, why are we doing all this? Well, we're trying to understand what it means to be healthy. How does that change over time? How does that differ between different people? What happens when people first get ill or wind up in a strange environment? And lastly, can we use these advanced technologies to better manage people's health? So can genome sequence really be useful for you? The time we started this, that was actually rather controversial. Most physicians would say, absolutely not. You should not be sequencing people's DNA because you'll turn everyone into hypochondriacs. So anyway, getting at this last point, can these advanced technologies be useful? Well, it turns out from the first, say, 3.5 years of the study, we actually found 49 major health discoveries, 67 if you count hypertension, for these different groups. So nearly half the people learned something important, and it's a wide range of different areas. So it's metabolic, cardiovascular disease, hem-oncology, what have you. All these different areas we discovered something, and in some cases it was a pretty big deal. So you'll notice in the hem-oncology, one person we caught had early lymphoma. I should say all these were found pre-symptomatically. And it was by doing these deep measurements. So one person had lymphoma. Right underneath that, you'll see MGUS and smoldering myeloma. Those are precancers that can convert to aggressive cancer. So it's useful to find those. In the cardiovascular space, we found 2 people with serious heart issues, one from genome sequencing, another from wearables, and so on and so forth. I'll go through some examples right now. And the key out of all this is that no one technology found all these things. It was a combination of all of them that actually uncovered these health findings. And so we would argue they all have value at different frequencies and at different expenses. So just to run you through a few examples, genome sequencing was powerful. We found 12 people with what are called Mendelian traits. So a famous example I'm sure you're all familiar with is BRCA mutations, put women at high risk for breast and ovarian cancer. And it turns out that there was an individual who had a BRCA mutation. There's quite a few things there. 3 of them actually turned out to be really quite significant. One person, fairly young fellow, actually had a mutation in that RBM20 gene, which is a cardiomyopathy gene. And it turns out a follow-up test, well, it turns out his father had a heart attack and died in the 60s, and his aunt had one in her 50s. And it turns out he did a follow-up stress echo after we sequenced his genome, and sure enough, he's got a problem, and he's actually on medication now. He did stress echo. Another individual had a mutation in the SHDB gene, which puts him at high risk for certain cancers, did a whole body MRI follow-up, and actually had early thyroid cancer, and that part was removed, and they were able to maintain their thyroid and then don't need thyroid replacement therapies. Another individual, there were 9 individuals thought to be type 2 diabetic coming into the study. One of the individuals actually turns out they have a MODY mutation. It's the second one from the bottom, that HNF4 case. That actually is a different kind. It's not a normal type 2. It's a very different kind. And actually usually treat them differently. And this person switched the medication. They've been on the suboptimal medication for years. But once they learned this, they switched. There's another person who learned, not shown here, something pretty important about their pharmacogenomic variant. They were on a medication, put them at high risk for heart attack, but they actually had a second one while they were on that medication. Then we sequenced their genome, they switched. It's pretty apparent they were on the suboptimal one. So anyway, genome sequencing was powerful. And now more recently, I won't have time to go through this, we've come up with new ways using machine learning for analyzing complex disease. And actually we can predict severe COVID with some reasonable sort of Framingham level prediction levels for those who, not age related, but the younger ones who actually get severe COVID and require hospitalization. I won't have time to go through this. I'll show you a few other examples though. This is the lymphoma case I told you about. This is a case where the individual had an enlarged spleen, but also had some elevated blood markers, and the combination tipped us off. At the bottom is an individual that had, actually there's 1,000 measurements there, and one individual had way too much IgM. His 2 dots, also a fairly young person, were just way higher than everyone else. And it turns out follow-up tests indicated he had way too many IgM producing cells and had this MGUS, this precancer condition. Again, and he'll be screened more. I have my own theories. But I should point out, I'm not an MD, so you can disregard everything I'm going to say. I'm also very conflicted. We form lots of companies, so everything I'll tell you is probably conflicted as well. All right. Anyway, other things we learned, 6 people had plaques in their arteries. I'm one of them. That's not mine, but mine looks pretty much like this, a little bit of plaque. And what you do there is take increased statins, and in my case seems to be going away. So that was effective. We're very interested in type 2 diabetes because it turns out I'm type 2 diabetic. I'm an unusual one, as you'll see in a minute. It was predicted from a complex disease phenotype by a Tool Butte's lab. And coming into the study, I mentioned 9 individuals were thought to be diabetic. Well, it turns out 2 were diabetic and didn't know it. So that was nice to find. And of course, a lot of people were pre-diabetic, that orange bar in the middle. And that made sense because 9 out of 10 people are pre-diabetic and don't know it. And if you haven't been measured, I guarantee a lot of you are pre-diabetic and don't know it because it's just very, very common. Why is that a big deal? Well, 70% of pre-diabetics will become diabetic in their lifetime. So we would argue it's important to know it. And I can tell you a test you can do right now. Well, not quite right now, but that white rice spikes nearly everyone. Okay, and if you had a glucose meter, you'd know right now whether you're spiking away badly or not. Anyway, we're very interested in how do people become type 2 diabetic, right? Do they just gradually get there? Does something trigger it? How does that work? Well, it turns out during the study from the first 4 years, 9 individuals became clinically diagnosed as diabetic and others had diabetic range values. And we asked for those 9 individuals, how did they get to type 2 diabetic? And the answer is 2 of them gained weight, sort of a classical way, gained weight, their microbiome diversity dropped. They became diabetic, went on medication. But for 7 other individuals, they actually didn't gain weight. Their weight remained constant. 5 of the 7 individuals gradually became diabetic. They're either the fasting glucose, hemoglobin A1C, or something called oral glucose tolerance test went up. And so they gradually got there. But for 2 individuals, something spiked them. And I'm one of those 2. I'm the top person there. That's 8 years of data. And orange is glucose and gray is the hemoglobin A1C. I think I have that right, hard to see from here. And so in my case, you'll notice I was running along fine. Suddenly there's that first spike on the left. That came right after a nasty viral infection. And what I did was, in fact, I was clinically diagnosed as type 2 diabetes. Looks like a small blip. That's about a year of data there. And I actually changed my lifestyle. I changed my eating habits, cut out all sugar. I watch what I eat even when I'm here. And I basically doubled my biking and started running, and I brought it under control. And it was so boringly low, I stopped looking at it. And then ironically, I looked at it later. See that 7.1 peak? And we don't have pointers here, but if you look at the top where it hit 7.1, that's when someone else looked at it and said, Mike, you're back to diabetic again. And I said, oh, I had no idea. So I sampled me a few times, and guess what? I'd stopped running. Guess when? Right where you see things go from normal to that second transition peak, and also had another viral infection. So it's not clear what triggered it, but I did start running. I brought it down, but never down to normal baselines. And then gradually it kept going up over many years. So that, by the way, that's, as I say, 8 years of data. And finally I said, all right, when my hemoglobin A1C got to 6.4, which is borderline diabetic, 6.5 is the official classification, I said, it's time for metformin. And it turns out I'm a non-responder, so I kept going up. And I should say before that, I tried something, which is I switched from being a runner, because I was running 4 days a week, about 4 to 5 miles a run, felt that was as much as my schedule could handle. So I switched from running to weightlifting literally 5 years ago, because the idea is that muscle mass is better for glucose homeostasis. That failed miserably. You'll see the line keeps schlepping up there. But I kept it up anyway because it's good for my posture. So anyway, so I do keep that up. And then I went on metformin, and that failed. And finally, what was the solution? Well, it turns out more data. It turns out that actually I'm insulin sensitive, which is a little unusual for type 2 diabetic. That is, my cells respond to insulin. Also make insulin just fine. So what's wrong with me? I don't release my insulin from the pancreas. And so there are drugs for that. Rapinolide, it's called, and I take that, and that actually works pretty well. Right now I'm on an SGLT2 inhibitor, and that works quite well. So the bottom line is additional data, and a lot of this data is trivial to get. You could actually get it on a lot of people pretty easily and better diagnosed. We call this precision diabetes. By better understanding what's wrong with you, you can actually tailor your treatments to that and hopefully have better outcomes. So anyway, that's the diabetes story. Now, one of the most important things we learned from this profiling study is shown on this slide. It's probably the most important thing you could learn, which is that we're measuring people first thing in the morning after fasting. And it turns out those profiles are quite stable over time. So this is just showing 12 people with 10 or more healthy visits. In my case, every dot's a different visit and every color is a different person. I'm the light blue person. And you'll notice that no matter which ohm you use, even the transcriptome, if I showed a third dimension, people will cluster together, meaning their dots will cluster together. So I'm light blue. You can look at the red one. If you look, probably easy just to look at the cytokines or clinical labs, you'll see each color, right? The dots are together. They're all measurements from the same person. For me, again, that 6 years of data, my dots are all together. They're not that much intermixed with other people's. Not only that, if you do a perturbation, we've had people do exercise runs, fiber treatments, that's kind of a lot of fun, viral infections, you will shift your profile, but you will still look more like you than anyone else. That is a big deal. This shows it here for exercise. Notice the color dots are all together. People ran to the VO2 max, half their molecules significantly change, but they still cluster together. Why is that important? Well, this is why you need to know your baseline. It's very easy to tell when you're getting ill by comparing a disease you, a perturbed you, if you will, from a healthy you, whether it's viral infection, in this case, exercise, I can show you, they're all the same. But it's hard to tell you, a disease you from a healthy you by looking at everybody else who's healthy. Does that make sense? The difference between people is greater than the difference between a healthy and a disease state. And that's why we think it's really important to get your personal baseline. And that's a hard concept for most GWAS folks to handle. And if there are any GWAS people here, I'm sure I just offended you, but it's true. Okay, so we think it's really important to get your healthy baseline and build personal trajectories. Now, I told you profiles are stable, but it turns out there are 2 times they shift, at least 2 times. And this is one of them here. This is seasonal changes. So we were thinking, well, we have all this data. How many biological seasons are there? It turns out, as you might imagine, right, we've classified arbitrarily the calendar year into 4 seasons, 3 months each. But if you think about it, who says there are 4 seasons? Maybe there's 15, maybe there's 3, maybe there's, I don't know how many. And I mean, it seems really likely they're not 3 months each, right? That's just totally arbitrary. So we could say, well, let's let the data tell us how many biological seasons there are. Now, these individuals we're profiling, they're all from Northern California, except for 3 from Southern California. And it turns out we can see, are there seasonal molecules that we're measuring? And out of the 20,000 molecules and many, many microbes we're following, we found 1,170 molecules and microbes that show seasonal changes. Okay, and it turns out that some of these are known already, most aren't known. Okay, and there's some interesting ones there. There's a pair gene that's involved in circadian rhythm. So we can say mathematically, how many clusters do these fall into? There's over 1,100 molecules. How many patterns are there? And the answer is there's 2. Okay, so we would argue there's 2 biological seasons in Northern California. And again, you can show that mathematically, 2 major seasons that most molecules will fall into. You'd say, well, there's spring, one's probably summer, one's probably winter. Okay, you'd be half right. So the one on the right, the orange pattern, those are molecules and microbes that actually peak in winter. See, the January and December, they're very high. And if you look at what's in there, it turns out, well, things like acne, this acute phase stuff, that's actually all viral infection related. And that makes sense, right? People get sick in winter normally. But the one on the left actually turns out not to be summer. In hindsight, it makes sense. It's late spring. Essentially, it's late April, early May. And if you ask what's in there, well, you'll see allergies and asthma, and that makes sense. But you'll also see other things like cardiovascular disease, diabetes, other markers that are there. And our explanation is that what we think is happening is even in California, people are less active in winter, right? It gets cold here and it rains, and for us, cold is 60 degrees, right? We don't like that. So the reality is we think people are exercising less. And then when spring comes, they get out there and do more. And so these markers go away. And there's things I don't understand. You can see seizures sleep markers actually peak late spring, early summer as well. So the bottom line is there's some good stuff in there. We get others that's not. Now we think this is important because some of this information has clinically relevant markers in there. We always like to tie things back to the clinic so that people can actually identify why is this important. So there are a lot of clinical markers that show seasonal patterns. Some of this was known already, but a lot of it's new, and certainly most of it's higher resolution than had been known before. And so we think this is a big deal because it lets you track these things if you're following your seasonal patterns, or if you incorporate this into the way you think about things, you're better able to, if you will, manage your health, knowing that, you know, if you're way out of whack compared to what you should be for a seasonal pattern, you might adjust accordingly. Or if you're high and it's late spring, you might know that you'll probably come down. The bottom line is you can incorporate this thinking into your medical actionability, if you will. The other time relevant to today's session is that the other time molecules change is over time, which we're interpreting as aging. Okay, and it turns out that we think you need as few as 5 measurements over 2 years, and we can tell if people are aging. So we're measuring these all from the same person, remember? So instead of cross-sectional studies, we can actually see how people are aging, and it turns out everybody's aging differently. I'm the one on the left, that's 4 years of data. I'm a pretty typical ager. My coagulation pathways, metabolic pathways, most of them go up over time. But the one on the right, their top pathway that's changing is actually a cardiomyopathy pathway. And in fact, we think then they're a cardioager. So we can take all these patterns. There were 43 people that had enough data, we could group them. Same thing, how many different patterns do we see of people aging? And the answer is there's 4 major ones. Now I know there's more than 4 aging patterns. We call these ageotypes, by the way. It's just that we're only statistically empowered to discover 4. And the 4 we discover: kidney, liver, metabolic, and immune agers. Okay, so we can see how people are aging, and we can classify people according to their ageotype. So every column is a different person. Every row is a different ageotype. And so the 4 people all the way to the right, they're aging in all 4 categories. They're getting older in all of them. The first box on the right, actually that person's aging in 3 of the 4, not so much of a kidney ager, but aging in the other categories. And let's see, I can't see, I'm somewhere in the middle. I'm not much of an immunoager, which I think is good, but I am aging in metabolic and the other kidney, liver. And the person I think all the way to the left that's boxed, they're a kidney and a metabolic ager. Next box over is a kidney ager. So you can see how the people are different, aging differently. And not only that, there are clinical markers associated with the ageotypes once again, so you can have markers that you can monitor, although we would argue it's best to monitor the whole ageotype. And what we find is that, I'll just run through a few examples. If you look at the bar with the star, that's the group as a whole, how they're aging. And if you look at the far left, that's hemoglobin A1C. On average, the group is going up. You see that bar going up. But if you look at the dark bars, those are individuals. 4 people have dark bars going up, and that means they're following the normal pattern. But 4 people have their dark bars going down, meaning they're reversing their hemoglobin A1C. And in this case, this is easy stuff. 2 of them underwent diets. I think 1 did exercise, some lost weight, and so on. So we have an explanation for why those went down, and that's an obvious intervention. Now, in the case of creatinine, which is a marker of kidney function, look at that, that one's in the middle. The group as a whole goes up, but we had 10 individuals going down, as though they're basically lowering their creatinine. And it turns out 8 of the 10 are on statins, and I don't think they're losing muscle mass. So they've somehow reversed their kidney marker, and there's more to discover there about what's going on. So the point is we can follow people individually for how their individual markers and the whole ageotype changes. So what do we think is going on? Well, we think of aging as like a car, right? Your car as a whole gets older over time, but some parts wear out first. Your immune system, your kidney, in one case, it's the cardio. I forgot to say the person that's a cardioager, we later learned was stage 2 hypertensive. So that sort of makes sense that they might be a cardioager. And so again, we think that by discovering this, you can actually take actionability. If you're a liver ager, maybe you won't drink quite so much wine tonight, I'm not sure. But the point is you can actually use the information in an actionable fashion. And lastly, I'll just mention that we don't have, I'd love to have the methylome on all these folks. We're actually going to do that, but it's really expensive. We want to do the whole genome or whole methylome, yeah, whole genome bisulfite. But we do have enough markers to do the phenoage stuff from Morgan Levine, and we can see how people are different aging. And so the chronological age is on the x-axis and the biological age is on the y-axis. And no surprise, people are aging differently. And of course, if you're on the left side of that diagonal line, you're older than your chronological age. And a lot of them have their slope going in a not favorable direction, meaning a pretty steep slope. And the people on the right are actually a better biological age than their chronological age. So we can see this now. I don't know that this is the best marker for aging. I'll probably offend somebody here, but anyway, it shows that you can follow people and it sort of fits with what others would say. So one question I get a lot is, how are you going to scale this? How do you get this out to millions of people? Well, I'm from Silicon Valley, so you form a company. And so we formed a company, Qbio, and they're doing a medical version of what I just told you, because what I showed you is pretty researchy. Doctor would know what to do with RNA-seq data, but we can actually do a medical version of this. And we have whole body MRI. And right off the bat, this is just from their first 100+ people, we found a lot of pretty important things. And it usually involved multiple measurements that actually tipped us off, in all cases, pre-symptomatically, once again. And in some cases, it was the longitudinal data that was key. You could actually see changes. Now, most physicians will tell you, don't do a whole body MRI. If I do a whole body MRI on every person in this room, I will find nodules. And that freaks doctors out. But that's not the point. The point is, do you have any growing nodules? That's what counts. So you need longitudinal measurements. And so you can see this while you're doing this. And speaking for myself, I have several nodules. Again, we all do. I have one on my spine. But if I got cancer and then got scanned, you would say, wow, you got a nodule in your spine that's probably metastasized. But it's been there. Well, I've been profiling me for 5 years. So it's been there. It hasn't been growing all 5 years. So it's kind of nice to know it's there. It's kind of nice to know it's not growing. And so that's the key. It's getting the right interpretation around these measurements. All right. We're doing a lot on wearables, and this is the last part of what I want to tell you. Wearables are quite taking over, so to speak. There's a lot of different devices out there that do lots of different things. And what's powerful about them is they can measure you 24/7. Now, smartwatches in particular, they measure minimum a few 100,000 measurements a day. The good ones actually measure 2.5 million measurements a day. And they'll measure you, once again, 24/7, 365 days a year, as long as you keep them charged. Okay, I'm a total believer in this. You can probably notice I'm wearing 4 smartwatches. I forgot my sleeve, so I'm holding my exposometer. We'll get to in a minute. I have a glucose monitor right here. I use 8 of these devices every day. Okay, so what do? Well, they let you measure all kinds of things, your activity, your heart rate variability. If you look at the bottom, respiration, they can measure all these different things. I won't spend much time on this because I want to talk about glucose and exposome, but we actually can use these to find infectious disease. In fact, the way we figured this out is I figured out my Lyme disease from a smartwatch and a pulse ox, because my heart rate jumped up while asymptomatic and my blood oxygen dropped. And then later went on and tested, and sure enough, I was Lyme positive. 2 weeks earlier, I was in rural Massachusetts, helping my brother put up fences. That's where I got it. But the bottom line is it tipped us off. And then we actually looked at all the data, 2 years of data time. And every single time I was ill, we could pick it up by jump up a resting heart rate in advance of symptoms, when symptoms were reported. And so we wrote algorithms to do this sort of thing. And I was ill 4 times, one of which was asymptomatic. And we call the algorithm change heart. The skin temperature, it will shift too, but that doesn't work well in everyone. So anyway, we can tell when you're getting ill from a jump up a resting heart rate. Top 4 are me with 3 other people, one of whom got sick twice. Same thing, worked every time on them. So I won't get into this, but we've now written, I'd love all you to join the study, so I'm going to make a shameless plug, but we've written algorithms to do this for COVID. First, we showed it worked for COVID. Now we have an alarming system where we can actually ping you in advance with an alert that you've got to jump up a resting heart rate. And it doesn't mean it's COVID, it could be COVID. It turns out it works for the COVID case, works 80% of the time, with a median of 3 days before or at the time 80% of the time before at the time of symptom formation, with a median of 3 days prior to symptoms. And it works on asymptomatic cases 14 out of 18 times. So the point is we can see this jump up from resting heart rate. As I say, I'd love to have you join our study. It's not just COVID, other illnesses will trigger it, and other sorts of activities can do it too. Actually, mental stress will trigger this as well. And we're trying to separate that up with more data. That's why we want you to join our study. Okay, other things you can learn from smartwatches are all kinds of things. So we can get some measure, this is from machine learning data. We're actually able to tell people's hemoglobin levels, hematocrit, things like that, which is an early indicator of anemia. It's not clinical value measurements. Don't get me wrong. You wouldn't use this for a clinical test, but it could tip you off if you're seeing a shift in any of these. Using our algorithm, you could see whether you're shifting and at lower levels of hemoglobin. And likewise for hemoglobin A1C, as well as fasting glucose, you can pick that up with a smartwatch measurement as well. It's a combination of features. And some of these features make a lot of sense. Actually, smartwatches can measure dryness of your skin. It's called your electrical conductance, ESR, if you've heard of this. So you can actually tell, it also tells stress, because when you're stressed, you sweat usually. So you can make all these measurements, and some of these things don't have clinical correlates. And so again, these devices can do things that you might not normally be doing, but still be powerful for your health. So I wanted to tell you a little bit about some of our glucose monitoring work, because Elise asked me to do this. So anyway, these things have been put out for type 1 diabetics and insulin dependent type 2 diabetics when we got involved in this. And we said, well, these are pretty cool. Let's put these on normal people and pre-diabetics and a couple of diabetics and see what happens. And what we discovered is that some people have pretty good glucose control, like the individual on the top. The individual in the middle is actually a moderate spiker. And then there are people who are thought to be perfectly normal who are spiking up the wazoo. Okay, they're just spiking just as bad as diabetics. So we've written algorithms to do this. I won't get into this, but we can basically classify these spikes and classify people into glucotypes, which is based on a combination of steady state and spiking levels. And what we've discovered is that normal healthy people can be either in a low, moderate, or a severe glucotype, meaning they're severe spikers. And so we've gone on to run people through different foods. And what we've discovered is different people spike to different foods. And Aaron Siegel actually has similar data, actually is a nicer study than ours. And basically, so we'll find that some people spike to bread and peanut butter, other people spike to protein bars, some of you will spike to pasta, others to bananas. It just varies a lot. And the microbiome is one component of this, but it's not the whole story. And so different people are spiking different foods, and these devices are really powerful for seeing this. And it turns out nearly everybody spikes the cornflakes and milk, by the way. So 80% of our folks do that. I think this is probably worse than cigarette smoking. It's not good for you. I hope they didn't serve that here this morning for breakfast, but muffins aren't so good either. And I guarantee nearly everybody spikes the white rice too. So that's not so good for you. Yeah, talking about sugar bans, I think some of these things you might consider as well. So this is a complicated plot, but what this shows, we can classify people according to their spiking patterns. And what we'll find, the so-called glucotypes, and what we'll find is that there are people who are normal by fasting glucose, hemoglobin A1C, oral glucose tolerance tests, and they're spiking just as much as diabetics. So it's missed, and I think that's because people have glucose dysregulation in different ways, and we're actually showing this now in other ways. Type 2 diabetes is probably 50 diseases, okay, all lumped together by high glucose. But the way you get there is very different. I'm not a normal type 2 diabetic. Most people will tell me. So the point is, it depends on your underlying mechanism. And I think that's what we need to tease out better. And we have new ways of doing this. We can tell you we've now run a recent study, totally conflicted on this one through my company called January AI. It's a metabolic control company. What they do, we ran a study with about 1,000 people, for which we got a fair amount of data. Once again, we could show normal people were spiking just as bad as diabetics, not as common in this particular study. But what's kind of cool, if you put one of these continuous glucose monitors on you, the same one I'm wearing right now, it turns out just wearing this monitor, you will get an improvement in your glucose control, which we can show. It's called time in range, time you're in the proper range of your glucose spike. So nearly 90% of normal people will improve their glucose just by wearing this thing for 10 days. Because if you've ever worn it, have any of you worn the continuous glucose? Yeah, a few. they're pretty absorbing, right? As soon as you put this on, you'll see what spikes you, and it always surprises you, and it always makes sense in hindsight. Like you think you're eating something healthy, then you put some dressing on it, suddenly you'll go through the roof, and it turns out that dressing is sugar, and you didn't realize that. So they're very powerful devices, and it's in hindsight, it's very obvious just wearing one of these things for 10 days will improve your glucose. The last thing I want to tell you about is the exposome, because I think this is something that's not measured very often. In fact, if you think about it, if you ask yourself right now, what are you being exposed to, both good and bad, you have no idea, right? Because we don't measure that stuff. Yet we know it's very, very important for our health. And so if you think about it, there's both biological and chemical exposures out there. And wouldn't it be nice to know about both? But so some of the basic questions we've gotten at is, how big is the exposome? How dynamic is it? How does it differ? How does it impact your health? But so a number of years ago, we started measuring this. We're just measuring airborne exposures right now using an exposometer shown on the left initially. This is the thing I'm holding now, is the second edition. We have a third one just about ready to come out. And so what we've done is actually use these to measure airborne exposures under the intake valve. They all measure PM 2.5, temperature, humidity, things like that. Under the intake valve, we have a filter that collects all the particulates, so all your biologicals and other things that might capture, bacteria, things like that. And under that's a chemical absorbent. And so we'll measure all the biologicals by taking those filters and just sequencing as deep as we can for RNA and DNA. And we'll measure your chemicals by basically taking the absorbent and doing mass spectrometry and seeing what chemicals got captured. And so what we discovered, well, this was the initial study we did. So 3 people, I'm the one at the bottom. I've been using these devices now for about 8 or 9 years, but we had 2 years of data for this first study, a little over. And then another person had it for a year, and another person for 3 months, and a few people for various targeted studies I'll tell you about. And so it turns out pre-pandemic, I used to travel all over the world so I could capture exposures all over the world. One of the other people is mostly Bay Area oriented, another travel a little bit about around the country. So we looked at people's exposures, and what we discovered is that, first of all, the exposome's pretty vast. Every single sample, in my case, I collect one set of samples during the week, one during the weekend. If I go somewhere special like here, I set up a special exposometer just to see what we're measuring now. Okay, so basically every sample has about 300 species in it. And it turns out the exposome is very dynamic. So we can group these. This is just at the kingdom level. This is looking at fungi, which is yellow, plants are green, bacteria are red, and then animals are blue. And so what we've discovered, I'm P1, there's another person P2, another person P3. It varies a lot, depends where you are, whether you get a fungal, bacteria, what have you, exposure. Okay, you can cluster these things down below by the type of exposure. And you'll see for me, the green, there are some days that are mostly plant, or some periods that are mostly plant, others that are fungal, others that are bacterial, and so on and so forth. So you can then say, well, what's your exposome mostly due to? Is it location? Is it season? What is it? And the answer is location definitely drives a lot of this. This is just showing mine, but same for the whole group. You can start teasing out patterns of location. We can quantify this. How much is due to location? That would be the spatial part. How much is environmental? That actually turns out to be seasonal. There's a lot of it we can't explain. I actually think it's location as well, but it's hard to quantify the way we do this. Okay, so we think location's a big driver. So we can actually test this. We took 4 people who were actually all living roughly in the same area. One's me, I'm the red dots this time, and I actually travel a lot back then. And so my dots are a bit scattered. There's a person who works in San Francisco, that's the blue person there. There's a green person in Sunnyvale, and the red person lives and works across the bay. So it turns out we can look at their exposures, and you'll notice that they're all different. So even though we're in roughly the same area, our exposures are very different. And the person in San Francisco, you know, again, theirs is different. We can see what organisms are driving this. Well, in their case, you know, you see all kinds of things. Some of them are kind of interesting. The one in San Francisco has sludge bacteria in their exposures. And if you've ever been to Market Street, this makes a lot of sense. But that probably only works for the locals. But anyway, the point is you get very, very different exposures depending where you are. Okay. There's also a strong seasonal effect. It turns out we have hundreds of organisms that show seasonal patterns, and I've just grouped them here, most of which were not known, actually, at least we didn't see them in the literature. And so many things show seasonal patterns, and I'm showing the top ones, which make a lot of sense. There's the very top one there, I think is pine. I'm looking at it right. I guess I should just look here. It is pine. And that, of course, peaks basically that's late spring, early summer. And then the fungus one at the bottom, actually, it's an autumn, basically, exposure. And so that comes up then. So you can actually see where these different things show up. And you can see there are pathogens, and these are very low level, though. We picked up most of them in New Orleans, interestingly enough. So anyway, what about chemicals? Well, it turns out there's a lot of chemicals in all our exposures. We picked up 3,000 different chemicals, we think. Most of them aren't identified. It's tricky to do this with mass spec, but at least 158 of them are known. And it turns out, if you look at what's in there, DEET is in all the samples, so that's stuff used to keep insects off. Certain carcinogens are actually in virtually all the samples, but they vary a lot from location to location, as I'll show you in a minute. Plastics are everywhere, so on and so forth. So we can measure all this stuff. And then we'll see which ones have interesting patterns. Some of these are seasonal patterns, things that start low and go up, or the other way around. So we can see that. We also have things, this is one that's influenced by location. This particular set is actually also influenced by the weather. So it turns out you probably know you can smell when it's going to rain. Do you guys know that? Because of this molecule called geosmin, that earthy smell molecule, that actually the air pressure drops and that comes out of the ground. So that's how you know when it's going to rain. And it turns out those same periods, whenever you see that thing going up, you'll notice plastics go down, body scent goes up, either because people are indoors or maybe it's coming off of them more, and pesticides come out. So we think they're probably coming out of the ground too. Plastics go down, maybe they're getting washed in, not clear. We can ask what kinds of correlations are there between the biological and chemical exposures, and there's tons of correlations on the external exposures. So especially you can ask which guys are the most connected. Well, the plasticides, plastics are highly connected to the chemical exposures that you're exposed to. And so we can start correlating chemicals and biologicals. And so we have something like 69 of these correlations. And this is one kind of cool example. So this is correlating basically fungi with a particular organic called pyridine. This used to be found in paints. It's only recently been outlawed, but it's still in a lot of other areas. And so it turns out wherever you'll have pyridine, you actually don't have much of fungi. So if you look at the pyridine peaks from the bottom, they anti-correlate with fungi, okay? So why is that a big deal? Well, my house was painted by a green guy, no pyridine. Every time I go home, I get a fungal exposure. But you can go to other areas where you'll see they actually have bacterial and other fungal exposures that are probably suggestive of perhaps I misspoke. Anyway, you get the idea. If you have pyridine, no fungi, and vice versa. And so what you might think is, well, maybe if you're allergic to black molds, maybe you want to paint your house with a pyridine paint. Might die of cancer, but at least you won't be sneezing all the time. Okay, so anyway, these are life choice decisions you can make based on your exposures. One thing you can ask, all right, how much of the external exposure gets inside of you? And so this wasn't the most sensitive assay, but we were able to detect of all those chemicals on the outside, the 158, we found 11 of them showing up inside us. And we found others as well that we didn't necessarily detect on the outside. Actually, 67 known environmental compounds we found in this case, it's on me, in my blood. Okay, so we can see these. But more importantly, we can start correlating external exposures with internal effects, whether it's a direct one or not. So to show you a few examples here, you can say, well, how does your gut microbiome, very few of the microbes outside you ever wind up inside you. Okay, they just don't get through your stomach and get in there, but they can still have a big effect. So you can start correlating external exposures with internal gut microbes, if you will. So these, what's circled there are the gut microbes that are highly connected to the external exposure, which is on the outer circle. And that's a combination of chemicals and microbes. And the point out of all this is that you can start seeing what kinds of gut microbes or correlated with what kinds of chemical compound exposures that are out there. And so it turns out that these pro-inflammatories are correlated, as you might imagine, with some of the things I've listed there. Plasticizers, I think it's, what else is in there? Pesticides, plasticizers, and the PPCPs will all correlate with these pro-inflammatory. And the one anti-inflammatory marker in the bottom right, that one is positive, it's negatively correlated with the external chemicals. So you can see how these certain chemicals are associating with your immune system. Does that make sense? So it's kind of cool, actually. Then this is Exposome, proteome, same thing, because we're getting late and I want to save time for questions. You can start correlating your exposure with other markers, in this case, protein markers. And it turns out, I think the top one here is, once again, it's a plastics. I forgot what the top categories here. Anyway, they correlate with immune molecules. And then we can get correlations between your exposome and your metabolome. And that'll relate to kidney and liver functions. And lastly, you can start correlating. Medical folks always like us to correlate things with clinical markers. You can see what exposures will correlate with relevant clinical markers like eosinophils. This one makes a lot of sense, glucose levels, things like that. So we're starting to make these connections. We obviously need to do this on a bigger scale. So this is a ton of data. How do we manage it? Well, we actually built a dashboard called My Personal Health Dashboard that lets you bring in your wearable data, your microbiome, your clinical. We don't have it set up yet for the Exposome, but we are working on that. And you can look at it, whatever resolution you want, daily, weekly, monthly, what have you, to make your own personal correlations. And then you could share this with your physician as well. The way we built this highly secure environment, can share it with physicians. And this is what I view as Mike Snyder's world. I envision a world where we're getting our genome sequenced before we're born, obviously not for us now, but for the future. And then measuring multi-omics measurements together with wearable information could let us better manage our health. So I've been very fortunate to have an amazing group of people in my lab, and I've just listed some of them here. Great collaborators with Tracy McLaughlin, George Weinstock, many others. We now have a very large wearables team all working on this full blast to try and make an impact. So I'll take questions if you have any.
Didn't I tell you? It's amazing, right? Gives a whole new term to me search. So questions, go for it.
I can repeat it if you have, maybe it's not on. Yeah, it might not be on.
There is a switch. Okay. First, I did January AI, so I'm
Great.
definitely in the space of doing these sort of continuous sensor measures. But I wanted to sort of pose the other side of this, and your sort of principal thesis was that we want more measures more of the time. And I'm just remembering an old talk that Bob Kaplan gave, where he sort of gave the exact opposite argument, saying that everything that we're measuring, we're doing a much better job measuring subclinical processes, and most all of them don't turn into functional decline, frank disease, or early mortality. And so his argument was we should be really thoughtful around this process because it's going to just cost a lot to do this. And I'm sort of wondering how you're finding the signal or thinking about that in terms of scaling this up of the economy of the whole thing.
Yeah, that's a great question. There's no question what we're doing is a research project, because I would argue we don't know what to measure. We want to discover new biomarkers with disease and things. I didn't get into this, but in the early lymphoma case, we see some very interesting cytokine markers going up prior to that. And I don't know if that's going to be a general marker yet, but it could be. And that's the kind of thing we hope to discover. So once again, this is research. And so most of what we measure probably will have no value. I don't think any of it goes to waste per se. We try and take what we think is most important and also cost effective. And that, to be honest, again, shameless plug, but this is what Qbio is about. It's not cheap. It's $3,500 to actually get one of those Q exams, as we call it. You may say that's a lot, but I would argue if you get a heart attack and go on long-term disability, that's incredibly expensive. So you can make economic arguments for people, certainly for cardiovascular disease risk, things like that, that actually pays to do this kind of stuff, especially at $3,500. And I think we set these up knowing they'll be expensive in the future. They should become a few $100s in my opinion. And so we're all working on new technologies to take the most useful information to exactly what you're saying, try and roll it out, but something way, way more than what we're doing now, which is a terrible way. I would argue we're in a world where there's 1,000, maybe 1,500, piece jigsaw puzzle about our health. And we're looking at like 5 pieces trying to figure out what the puzzle is. And I think we've got to get to the 1,500. That's how I view it. And I also live by the motto, there's no such thing as too much information. So as you could probably tell.
Thank you, Dr. Snyder. I'm Christy White. I'm a clinical health psychologist, and my area of interest is in climate change, sustainability, and health. And so one of the ways that this comes out in my clinical work is how can we make our clinical work more climate aware? And so I'm working with patients on how can we adapt their care plans when there's a climate related or environment related event. During your talk, all of these sort of dots were connected for me. And I'm wondering if you've ever thought about or considered partnering with environmental scientists or people doing climate change work to see if any of these environmental exposures are connected to environmental events and how that shows up individually for people who might be at greater risk for some of these climate related events due to their biology.
Yeah, that's a great question. Believe it or not, we are teaming up with someone in it's the Woods Environmental School to try and do some of that. And we're just so much at the beginning because we don't have but I think that would be fantastic to do. And I think we're really trying to drive the price down of these exposometers. This is the first one I showed you was about $2,500, not counting all the assays we do downstream. This one's $1,100, and we're trying to make one that's more like $150, so we can then get it out to thousands of people. And I think try and figure out the best way to do this. I think what you're raising is really super cool, something we're very interested in. So yeah, but we're not there yet, barely even starting.
Hi, I'm Beth Cohen from UCSF. Thanks for the really interesting talk. And I also had a question about thinking about moving this from screening to like the research screening to clinical screening. And obviously you want to balance what you presented, which were some really interesting clinically actionable findings that improve health, with what you mentioned with, the things that might be just false positive or abnormal, but aren't clinically actionable. And I'm curious about, in your studies, are you measuring that other side of, potential distress caused by this amount of data, or things that you find that don't actually lead to improvements of health? And how do you think about that balance in terms of finding the right population to apply this to, where you're maximizing the benefit and minimizing that risk?
Yeah, these are great questions. So that's really good. What we do is, first of all, we usually have multiple lines of evidence that will help show us what's going on, like the lymphoma case I mentioned. We saw the enlarged spleen, but we also see these other blood markers that went up in the combinations, and something's up here. And so then we obviously did more follow-up tests. So what these do is they kind of give you hints, especially when they're not clinically validated tests. Now imaging's getting closer, not the imaging the way we did it. So what they're doing is giving us hints that can have you do follow-up tests. And the concerned people say, well, won't that lead to millions of dollars of follow-up tests? And the answer is no, it doesn't actually. You have a pretty good idea what's going on. And I would argue even now, believe it or not, people go to a doctor's office and they'll have high glucose. And people write off, say, well, you had a cold that day, probably didn't count. Come back 2 years from now, we'll see. So even the way we do things now in the medical system is flawed. And I'll tell you another argument, which is, I thought was quite interesting for me. The normal range can be very large for a lot of these clinical measurements. So a lot of people like me, I'm very low, I can tell from my genome why, but I'm low on certain markers, my eosinophils. And then actually another marker as well. And then it'll be marching along just fine. It'll jump up, it'll be borderline, but still in the normal range for some of these values. And people say, oh, it's fine, you're still normal. But I'm normally so low, and this thing jumps up high, and do follow-ups, you say, yep, something's not right there. So it's really taking advantage of longitudinal information, I think can help eliminate some of what you were just raising. If we have multiple measurements, we can better see what people's trajectories are to try and get a better feel for what's real and what's not. So I think it's a combination of longitudinal data as well as the big data, the multiple lines of evidence, help reduce false positives. I think that's the key. And then how do you do this in a cost-effective fashion? That we still have to work on. Yeah.
Hi, thank you. My name is Quinn Conklin, and I am in the psychology department at UC Davis. And you had mentioned that early on in the talk, there was some hesitancy around giving people their data because you might turn everyone into hypochondriacs. And so I'm wondering if you have any, if you're gauging that, how people make sense of this information. And then that relates for me to other questions of accessibility and data privacy. And so you obviously have an incredibly rich framework with which to interpret all the data you're collecting about yourself, but how does that translate for people who might not have that sort of framework? And what sort of education are you doing with people to make sense of those data?
So great question. So believe it or not, so first of all, we are selecting for eager beavers to be in our study, right? You're giving tons of blood. So we're probably selecting for a certain type of individual, and they've all marked, we run surveys, I didn't get into this, they've all learned something important and improved their habits actually as part of being in the study. So there has been a positive. But believe it or not, our biggest problem isn't the hypochondriac. It's actually the opposite. People forgetting, like we'll tell them something really important, like you're pre-diabetic and you're borderline diabetic, and then they kind of go off and they remember initially, and then they kind of go back into whatever their old habits are, and maybe forget that they're pre-diabetic and they're eating badly and behaving badly. So we're not having the problem you described that one might have thought. And we worried about this a lot, and I showed you part of the study. We've done a lot with genome sequencing, and that was an early argument when we started sequencing healthy people's genomes. Physicians just hated that because they worried it was going to be. But the reality is there aren't that many follow-up tests that you wind up doing. So it's not that expensive to do those follow-up tests, about $700 a person. And the other is it just hasn't really been the big issue. Again, it might be because we're selecting for people. And I think the way to do it in the long run is people should be in charge of what they want to get back for information. So if you're an eager beaver, you want to learn all, that's great. But if you're maybe a worrier, maybe you don't want to get all the information back. Maybe you want it to go to your physician, but not you, so that they would actually do the right test when you go get checked up without you knowing it and catch things. What you don't want to do is have, you don't want somebody waking up every morning saying, I'm going to get breast cancer, I'm going to get breast cancer. That would be a bad thing. And likewise, you don't want, helicopter moms and dads over their kids saying, I worry about my kids because of all these risk factors. So I think you have to manage it appropriately. And try and put it in reality. But I think people are very capable of understanding this stuff. I think the medical establishment, in my opinion, tends to think we're the only ones who can really interpret this and relay it back. And actually, you'd be amazed at how good people are. In fact, that most people are way better than doctors, because doctors have 15 minutes to spend with the patients, right? Whereas they have eons to spend to look at themselves and get a better feel for what's going on. So I'm a believer in power to the patient.
Have you had any resistance from IRBs in terms of giving people their information?
Oh, the IRB fights us tooth and nails. They wouldn't let us give back the data for 7 years to the patients. And they would say, well, it's not clinically validated. Wait, RNA-seq is not going to be clinically valid. It's not clinically validated. Therefore, it's not clear, so you can't give it back. So we would argue, well, if it's not important, why can't we give it back, like your microbiome? And if it's important, shouldn't we tell them? So the whole argument made no sense. But after 7 years, it changed. And you know why? Nothing. They grew up. We just kept resubmitting every year that we want to return data to the patient. And finally, they approved it. We did absolutely nothing different.
Thank you.
Yeah.
Thank you, Mike. Fantastic. Okay, this is a bit of a marathon.